Hello students, how are you? I hope you will be fine. Students, in today's session, we will discuss about proteins, their properties, their structures and types of proteins. So this is for unit number 14, biomolecules. So let's start. In today's session, we will discuss about peptide bond, classification of proteins, structures of proteins. These structures are primary, secondary, tertiary and quaternary structure, and after that denaturation of proteins. So let's start. First we will see the peptide bond or peptide linkage. So students, as you know, we in my previous video we have discussed about amino groups. What are amino groups or what are amino acids? Okay, so you have studied there. In today's session, in peptide linkage, so first let's define peptide linkage. It's a bond. And which bond? It's a amide bond. Amide bond means CONH2, that is amide bond. So this peptide linkage is an amide formed between carboxylic group and amino group of two different amino acids. Remember this thing, in amino acids we have seen the structure of amino acids. In amino acid, one carboxylic group is there and one amine group is there. One hydrogen is there and also one R. So R may be different for different amino acids. So the compulsory condition is carboxylic acid and amine. When two amino acids combine, okay, so the linkage or the bond, it is formed between carboxylic group of first amino acid and amine group of second amino acid, and from carboxylic and amine one H2O is removed. So if H2O is removed, so from amine one H and from carboxylic OH, that is H2O. So the remaining part is NHCO. Okay, so that bond is known as amide bond. So what is peptide linkage? Peptide linkage, it is formed between amide which is formed between the carboxylic group and amino group. If two amino acids are bonded, the product is called dipeptide. Di means what? Di means two, peptide means amino acids. So dipeptide means if two amino acids are combined, it is dipeptide. Tetrapeptide, tetra means 4, tripeptide means 3, pentapeptide, okay, and so on. So what mean here? 2 means dipeptide, 3 means tripeptide, 4 means tetrapeptide, 5 pentapeptide, 6 hexapeptide. Hexapeptide means 6 amino acids are there. Okay, so when the number of amino acids are more than 10 are bonded, the product are called polypeptides. Like dipeptide, tetrapeptide, if the number of amino acids which are combining, if the number of this amino acid it is more than 10, it may be 100, it may be 50, maybe more than 100. So if it is more than 10, then it is known as polypeptide. Okay, and a polypeptide with more than 100 amino acid residues having molecular mass higher than 10,000 mu is called a protein. So now here we are going to define protein. What are proteins? So proteins, these are made by repeating or by combining the amino acids, and the number of amino acids it is more than 100. Okay, so that the molecular mass it will be more than 10,000 mu. So very simple fact, in protein, if you decompose protein or if you hydrolyze protein, then more than 100 types of amino acids molecule will be formed. Okay, so this is a protein. In very simple way, let's see the peptide linkage. Okay, so in peptide linkage, what can we how can we form the peptide linkage between two molecules? So see, this is the structure of peptide. Here is H, here is R, here N, H. Okay, so like this, one H, one H. Okay, here is COOH. So this is first molecule. Second molecule, the structure of COOH, it is like this, COOH, then carbon, hydrogen, R, and here again NH2. So from carboxylic 1 OH and from amine 1 H, they will form H2O. Okay, so H2O is removed. After H2O, nitrogen, its one bond is vacant now. From here one bond is vacant, from carbon one bond is, so here nitrogen and carbon will be bonded. So this will be the linkage, this type of linkage, NH, here is CO, and again repeating units. So this bond is known as amide, because formula of amide, this formula is CONH2. This is amide, one hydrogen less, so this is also amide. Okay, so peptide bond, it is actually amide bond, and this is formed between NH2 and COOH. Got it? Now, classification of proteins. So on the basis of their molecular shape, we can classify proteins as fibrous protein. When the polypeptide chains run parallel and are held together by hydrogen and disulfide bonds, then fiber-like structure is formed. Such proteins are generally insoluble in water. Example, keratin, present in hair, wool and silk, and myosin, present in muscles. So what are fibrous protein? See, suppose this is a amine group. This is amine group. So between amine, that is CONH, again amine, again NH, CO. So this type of repetition will be there, and this is the straight structure. This is straight structure. So when two or more the fibrous chains, they are run here, when polypeptide chains, they run parallel and held together by hydrogen and disulfide bond. Hydrogen bond means, here see, if we draw here between hydrogen and hydrogen bond, so hydrogen bond will be formed between these two. See, here is NH, here is CO, A, again NH, CO, and so on. So between hydrogen and this oxygen, hydrogen bond will be there. So this, due to this hydrogen bond, these two chains, these are bonded with each other. They are running straight. Okay, they are moving straight, but they are bonded via hydrogen bond. Okay, so this type of proteins are called fibrous protein, fiber like structure, thread like structure. Okay, second type of protein, this one is globular protein. This structure results when the chain of polypeptides coil around to give a spherical shape. These are usually soluble in water. Example, insulin and albumin. Okay, now see, in fibrous protein the structure is straight, but here what will happen? This structure will rise like this. So this is a spherical structure. This is a protein. Okay, this is definitely protein, but the structure is like globule, like spherical. Okay, sphere is sphere like structure. So when the polypeptide chains, they are coiled around to give a spherical shape, these are known as globular protein, and the example is insulin and albumin. Got it? Structure of protein. So now structure of protein, these are primary, secondary, tertiary, primary structure, secondary structure, tertiary structure, and quaternary structure. So let's see, first primary structure. So in primary structure, what will happen in primary structure? The proteins may have one or more polypeptide chains. Each polypeptide in a protein has amino acid linked with each other. in a specific sequence, and it is this sequence of amino acid that is said to be the primary structure of that protein. Any change in this primary structure or in the sequence of amino acid creates a different protein. What is the meaning of this statement? Very simple meaning. In my previous video we have discussed about the representation of the amino acids. We are using three letters: guanine, cytosine, thymine, many more protein amino acids, different amino acids are there. Okay, so when these amino acids are formed, these amino acids are formed, so the sequence of amino acid, this sequence is very specific. This sequence is very specific. Specific means what? The order of the amino acids, it is fixed for a particular protein. If you change this order, then definitely the nature of the protein, it will be changed. Okay, and also the type of protein will be changed. For example, glycine. So you see the symbol of glycine, it is GLY. After that ALA. After that VAL. After that LEU. So this is the sequence, fixed sequence. Suppose this one is giving rise to a protein which is A type of protein. If you change this sequence, glycine, ALU, and just LEU and VAL, if you just change the sequence, the type of protein will be changed. It's now not A protein, it's a B protein, second type of protein. Okay, if you replace one from this A, if you just change this valine to lysine, in place of valine if you write LYS, then again this A is changed to now C. It means, what I mean to say, if you change the sequence or if you replace the protein, that amino acid, so the nature and the type of protein will be changed. Okay, and also the properties will be changed. Got it? The disease which is known as the sickle cell anemia. The disease is sickle cell anemia. This is due to the change in the sequence of amino acid. One amino acid is replaced from the chain, and due to that lack of amino acid, the disease is caused. So this structure is known as primary structure. Here see, now these four proteins. So what will be the sequence? So what will be the structure of protein? So in a very simple way, here amino acid, I'm writing amino acid like this one. See, NH2 CHR, here again COOH. This is the amino acids. So again one amino acid, so here see, from NH2 one H is replaced, from carboxylic one OH is replaced. So what will be the structure? NH CHR CO, again NH CHR CO, again NH CHR CO, again NH CHR CO, like that. So this is the straight structure. This straight structure is known as the primary structure. Okay, nothing is special there. Simply we have to add amino acids in a sequence, so it will give rise the primary structure. Secondary structure. So in secondary structure, the conformation of the polypeptide chains assumed as a result of hydrogen bonding is called the secondary structure of protein. What I mean here, see, if this is a primary structure, so in secondary structure the coil, it will be like this one or it will be like this one. So what we have to do here, we have to just make the coil from this straight rope. Okay, so see, depending upon the size of R group, two different structures are possible. First is alpha structure, alpha helix structure, and second is beta structure. So let's see here in secondary structure alpha helix and beta or pleated structure. First alpha helix, you can see we are rotating it clockwise. Okay, we are rotating this one. So here you can see in this sequence, in this particular sequence, what will be the structure? It will be C double bond O. Then after this C, there is N, H, then CHR, then CO, then NH again. So this type of coiling will be there. Got it? What is happening here? Carbon, which is bonded with oxygen, and nitrogen, it is bonded with hydrogen. So between hydrogen and oxygen there will be hydrogen bond, and it is hydrogen bond because of which the structure is helical structure. Okay, you can see here. This is the complete structure. Here you can see CO, CO, CO and NH. So where is hydrogen bond form? Between CO and NH. This is hydrogen bond. This is the hydrogen bond. So this hydrogen bond is responsible for the attraction between these two coils, because this carbon is bonded with nitrogen. So this will be like this. It will not go away. If you break this bond, then it will be structured like this. Okay, otherwise it will be attracted. So simple. In alpha helix there will be hydrogen bond between carbonyl group, that is CO, and amine group. Okay, so between oxygen and hydrogen bond will be formed. This is the alpha helix structure. Second one is beta pleated structure. So in beta pleated structure, again see amino acid. What is amino acid? NH. After that CHR. Okay, after that CO, again NH, again CHR, after CHR CO. So this is the sequence. This is the sequence. Getting your point? Okay. So in this is the first chain. This is the first chain, and this one is second chain. So here two chains are moving parallel. Here is only one chain is there. Just we are coiling this chain. This is the coiling of chain. But what is happening here? In this case, one chain is there, second chain is there, and between hydrogen and oxygen, hydrogen bond will be there. Between hydrogen and nitrogen, oxygen bond will be there. So this type of structure is called the beta pleated structure. In this beta pleated structure, see, this is the third chain. So what is happening here? Here this is the structure, the seat like structure. As you seen, the seat which is used in our houses, in the roof of the house. So that seat has curved structure. Curved structure means like this. This is the seated structure. Okay, again this is the seated structure. So between these seats there are hydrogen bonds. Okay, this is beta pleated structure. After beta pleated structure, this is the secondary structure of protein. After secondary structure of protein, now we will see the tertiary structure of protein. Okay, so the tertiary structure of protein represents overall folding of the polypeptide chains. Further folding of the secondary structure, it gives rise to major molecular shape, fibrous and globular. The main force which stabilizes the 2 degree and 3 degree structure of proteins are hydrogen bond, disulfide linkage, van der Waals, and electrostatic force of attraction. So what is tertiary structure? In primary structure, this is the primary structure. Secondary structure, this is the secondary structure. Okay, and tertiary structure, so just you coil this one. This is the tertiary structure. What we are doing here, we are making this coil much more complicated. We are folding this coil again and again and again to give a particular shape. That shape is fibrous or globular. Okay. The main force which stabilizes secondary and tertiary proteins, structure of proteins, So these forces are hydrogen bond, disulfide linkage. Disulfide linkage means between S and S. This is the disulfide linkage. Okay, we'll see next. Van der Waals force and electrostatic force of attraction. So this is the tertiary structure. And quaternary structure, some of the proteins are composed of two or more polypeptide chains referred to as subunit. The special arrangement of these subunits with respect to each other is known as quaternary structure. It means now see, sequence of amino acid, it is giving rise to the formation of protein, but some proteins, they have two different polypeptide chains or some more different polypeptide chains. For example, hemoglobin, it has two types of polypeptide chains. Okay, total 4 polypeptide chains are there. So if 4 polypeptide chains are there, then definitely the structure will be much more complicated. So what can we conclude here? In a very simple way, we can conclude the structure of primary, secondary, tertiary and quaternary structure. Primary, very simple, straight, just add amino acid one by one. This is primary structure. Secondary structure, making coil. So this one is now getting more complicated. Tertiary structure, make coil, folding of coil, it will take place. Okay, so again you have to fold the coils, and after that last one, quaternary structure. So you can see now this one is much more complicated. Okay, and here two different types of proteins are there. You can see this is the first protein, first polypeptide chain. You can see now this one is first polypeptide chain and this one is second polypeptide chain. So when two polypeptide chains or three polypeptide chains are there, the structure is quaternary structure. Okay, so primary is very simple, straight, secondary coil, tertiary quite complicated as compared to secondary, and quaternary, it is formed when two different polypeptide chains are there. Okay, here only one polypeptide chain is there. Chain is there. Here we are just calling single polypeptide chain, but here two different polypeptide chains are. Okay, so these are the structures of protein. After the structures of protein, now we will see the denaturation of protein. So protein found in a biological system with a unique three-dimensional structure and biological activity, which is called a native protein. It means whenever we talk about protein, then structure of protein, it is fixed. Okay, structure of protein is fixed. That quaternary structure, secondary or tertiary, it is fixed. Okay, it's not compulsory that all the proteins have quaternary structure. Some proteins may have only secondary structure. Some proteins, they may have tertiary structure, primary, secondary and tertiary, and some proteins, they may have primary, secondary, tertiary and quaternary. So primary structure is must. Okay, second, tertiary and quaternary, these are optional. So the properties of protein, the properties of protein, it is due to the structure of protein. Okay, properties of protein, it depends on the structure of protein. If you change the structure, then the properties will be lost. Okay, protein will be there, but the properties will be lost. So we can say the property of protein, it is due to the three-dimensional structure and biological. Okay, so this is the native protein. When a protein in its native form, means biologically active form, is subjected to a physical change like change in temperature or chemical change like change in pH, the hydrogen bonds are disturbed. You know, in secondary, tertiary and quaternary structure, hydrogen bonds are there. Okay, so whenever a change is there, change like physical change like temperature, chemical change like change in pH, addition of acid or base, the hydrogen bonds are breaked. These are disturbed. Due to this, globules unfold and helix get uncoiled. See, in straight structure, the primary structure, there is no any hydrogen bond. But in secondary structure, coil is there, and that coil was due to the formation of hydrogen bond between carbonyl and amine group. So that hydrogen bond is responsible for coiling of chain. If you break that hydrogen bond, the chain will be uncoiled. Okay, and helix will get chain. It will convert into a straight structure. So if the structure is changed, the protein will lose its chemical biological activity. So this is called the denaturation of protein. So simple see, denaturation, naturation, nature. Nature means the character or the property of the protein. Denature means change in property. So change in properties of protein, that is known as denaturation of protein. And why this change is taking place? Due to the change in the temperature, pH, or some other addition of some other chemicals. Okay. Due to denaturation of protein, or during denaturation of protein, the secondary, tertiary and quaternary, these structures are changed, these are destroyed, but primary structure remains unaffected. It means, see now, this is a primary, this one is secondary, this one is tertiary, and this one is quaternary. Okay, so if we are changing the pH or we are changing the temperature, it will convert into this one, again into this one, again this one. So what can we conclude? During the denaturation of protein, only the primary structure remains unaffected and the remaining structures are changed. This is the most important line. Okay, underline this line. So two very important question: what is denaturation of protein and which structure remains unaffected during denaturation of protein? So it is primary structure. Okay. Example, the coagulation of white egg on boiling. You know, egg, when you boil that egg, the liquid part, it converts into solid part. So the conversion of that liquid portion into solid section, that is known as the coagulation. And this coagulation takes place when we boil it, means when we increase temperature. So which kind of change is there? Physical change, like change in temperature. So due to change in temperature, the white egg, it is converting into solid. Also curding of milk. So whenever you add acid into milk, it converts into curd. So this is the due to the addition of due to the chemical change, like change in pH. And why pH is changing? Due to the addition of acid. So very simple. Again, if you add acid or base or some other chemicals, or if you change temperature, then the protein, it will change, or the nature of protein will be changed. That change in nature is called denaturation of protein. Okay, got it? So these are the denaturation of protein. So that's all for today's session. Now if you have doubt, you can contact me. Thank you.